So let's look a little bit about how that is working. So as I told you before, we already talked about that the LAC operon contains a promoter. That promoter has the -10 and -35 region, and next to it and overlapping it, as you can appreciate here by these arrows, is the operator region. And eventually we have the expression of the LAC-Z gene. B5' of the promoter region, we have a CRP binding site where the cyclic AMP receptor protein can bind once cyclic AMP is available. So something that I want to also bring up to your attention is the fact that in the messenger RNA, we're going to have a Shine-Dalgarno sequence, which is a sequence that allows for ribosomes to bind to a messenger RNA and therefore translate that into a protein. So that usually happens about 8 bases upstream of the codon, of the start codon of the protein. And that's going to become a little bit evident later when we discuss the next time in class, how the Shine-Dalgarno helps produce proteins in a polycistronic mRNA. But anyway, going back to what we were discussing, the CRP protein binds specifically to the CRP binding site, but only when it is bound by cyclic AMP. So when cyclic AMP levels are low, CRP does not bind the CRP binding site. When cyclic AMP levels are high, it is then able to bind to the protein CRP, and together that dimer is able to bind to the CRP binding site. And that activates the promoter to be able to activate transcription. And we know that there's over 100 different operons that could be controlled by this mechanism of catabolite repression based on the levels of cyclic AMP and the CRP protein. So how is this happening? And this is an illustration from a thesis from Valentin Sulkover from the University of Grenoble in France, where it's illustrating some of the control of the LAC operon, including the binding of the cyclic AMP and CRP protein. So what we have, as we discussed earlier, it's a promoter region that is next to a operator region, and 5' upstream of the promoter, we have the CRP binding site. So 2 cyclic AMPs needs to bind to CRP, to a dimer, to be able to bind to the promoter. So now they're binding to the cyclic AMP. That's going to promote the binding of the RNA polymerase because the CRP protein is going to directly interact with the alpha CTD, the alpha C-terminal domain of the RNA polymerase, which now facilitates the binding of the RNA polymerase to the promoter, and that increases transcription. So when LAC-I has been bound to allolactose and it has been removed from the operator region that allows for the binding of the RNA polymerase also is going to be positively influenced by the presence of the bound CRP, and that is going to initiate transcription of an mRNA that can later encode for the beta-galactosidase gene as well as the permease gene to transport the lactose in the cell. Once the RNA polymerase begins to bind to the promoter and starts polymerization, the CRP protein is no longer needed, and therefore it can fall off. So here's an example again of the overall regulation of the LAC operon. So we have regulation at 2 different levels. The first one is the repression via the LAC repressor that is bound to the operator region, that is the protein LAC-I, and that can only be alleviated when lactose as an autoinducer is present. The other part of it is going to be the activation of the cyclic AMP and CRP protein that is going to only bind to the CRP binding site when cyclic AMP is available, and cyclic AMP levels are high only when glucose is low. So as you can see again in this image, we have the CRP binding site in blue, the promoter in yellow, and the operator region in pink. The operator region can be bound by the LAC-I protein, which is going to inhibit the promoter. As I mentioned to you earlier, the LAC-I has its own gene that is separate from the operon, producing the LAC-I mRNA and eventually the protein. The protein is inactivated by the presence of the inducer, and that's going to be allolactose. When the repressor is removed by the inducer lactose, as well as the cyclic AMP CRP dimer is bound to the CRP binding site, those two events positively influence the RNA polymerase holoenzyme to bind to the promoter and initiate transcription. And you're going to have now a polycistronic mRNA that encodes three genes, three proteins, the LAC-Z, the LAC-Y, and the LAC-A protein. And that helps with the metabolism of lactose. And that finishes our section on negative control of regulation.